Restriction enzymes. Restriction enzymes are also called molecular scissors. This is because they cleave the double-stranded DNA at or near specific sites. These sites are generally known as restriction sites. Prokaryotes like bacterial cells have oodles of these restriction enzymes. They use these restriction enzymes for their defense against bacteriophages and other invading viruses. Restriction enzymes prevent the replication of phage DNA. When the foreign DNA enters into bacterial cell, restriction enzymes snip it into fragments, but they do not harm the host's DNA. The host DNA has methyl groups attached to it, but the phage DNA lacks these methyl groups. Restriction enzymes cleave only non-methylated DNA. Therefore, they leave the bacterial DNA but break the phage DNA. Restriction enzymes are generally classified into three groups: type 1, type 2, and type 3 restriction enzymes. Type 1 restriction enzymes need ATP, S-adenosyl methionine, and Mg plus ions for their full activity. They cleave at 1000 base pairs apart from the recognition site. They contain 2 restriction subunits, 2 methylation subunits, and 1 specificity subunit. Type 2 restriction enzymes are the most widely available and used restriction enzymes. They do not need ATP but need Mg plus 2 ions as cofactors. They cleave at palindromic sequence which is 4 to 8 nucleotides in length. Remember, palindromic sequence remains the same when you read it in both directions. Type 2 restriction enzymes are made of a single subunit. They identify and cleave DNA at the same site. Type 3 restriction enzymes are made of more than one subunit. They recognize DNA at 2 non-palindromic sequences that are complementary to each other. They break DNA 20 to 30 base pairs apart from the recognition site. They also need S-adenosyl methionine and ATP for their proper functionality. Applications of restriction enzymes. Restriction enzymes are widely used in biotechnology to degrade the DNA into smaller fragments to study them. They are utilized in different experiments on gene cloning and protein expression. They are used in molecular cloning, DNA mapping, and gene sequencing, etc.
That is it for today's video. Make sure you are subscribed to the Science Info channel. Like and share this video if you wanna help others. Stay tuned. I will see you in the next video.